Thumbs up. We're starting. Great.
Hi.
Okay. Hello, everybody. Welcome to our Facebook Live session for April. I can't believe it's April because it feels like winter
Already.
outside.
So fast.
So, my name is Mary Sunderland. I'm the Director of Research and Education at the Foundation Fighting Blindness, but you can see we're not in the Foundation Fighting Blindness office right now.
Today.
We're in a much more interesting and exciting place. And we don't have our special microphones kind of set up, so if people can hear, will you give us a thumbs up or a hi online, and we'll make sure that our volume's okay. Because I'm joined by a very special guest, Dr. Brian Valios, and we're here in his laboratory at the University of Toronto. So Brian, why don't I pass it to you and you can introduce our setting a little bit more.
Okay, so I'm Dr. Brian Valios. I'm a Senior Research Associate and Resident Physician here at the University of Toronto, and today we've taken you outside the Foundation offices, and we're actually in the laboratory, and we're going to do a little brief tour of the lab, show you a little bit about the space where we work, talk a little bit about the projects we're working on to further treatments and cures for blinding eye disease, and then I think we have some very topical literature to discuss in terms of some big studies that have come out recently. It's been a very busy March and April for vision scientists everywhere.
Yes.
So,
we planned this event because such an exciting paper came out about the first stem cell clinical trial for wet AMD, and then like a week and a half later there was one for dry AMD. I haven't even had a chance to write it up. It's all hot off the press.
Yep.
So I'm really glad we can talk about it today. So stay tuned for the whole session. You don't want to miss it. But I thought let's start in the lab.
Yep.
And Brian has set up a little bit of an introduction to the retina. You know, we always talk about the eye, and we're talking about different kinds of cell types. So I thought it would help if Brian can kind of orient us. He's got an image set up on the computer over here, so we all know what we're talking about. So do you want to walk over here?
we're going to do a Facebook Live on the run.
All right, here we go.
So we've set up just a couple of slides to orient everyone. We talk a lot in the clinic with patients about the retina, but we never really get an opportunity to show patients what exactly the retina is. We talk about the retina as being like film inside a camera, but it's actually far more complex than that. The retina is layered like the layers of a sandwich, and those layers each serve very specific functions. And in this cartoon that we have outlined here on the retina, we see all these multicolored photoreceptors in this outer layer of the retina. And in diseases like macular degeneration, it's those photoreceptors which are lost and are responsible for the lost vision. The layer underneath the retina, the next layer of the sandwich, is the retinal pigment epithelium. And the retinal pigment epithelium is like the support cells of the retina. They support the normal function of the photoreceptors, and when those cells are lost in wet and advanced dry macular degeneration, it leads to degeneration of the overlying photoreceptors. And so our strategy in our research is to replace those cells in both of those layers of the retina. Our major focus in recent years has been replacing these multicolored cells, the photoreceptors, because we think that by restoring those photoreceptors, we'll actually be able to restore functional vision. In the studies that we'll talk about a bit later, most of the groups have been focused on restoring the support cells, the pigmented epithelium underneath. And those cells are also very, very important to try to rescue the photoreceptors. I've drawn here on this blue squiggly line the gel that we use to actually inject our cells into the retina. So that gel serves as a delivery vehicle for our cells, our photoreceptors that we want to transplant. It allows them to distribute in this space, and it actually supports their survival after transplantation to keep them alive long enough to integrate into the retina. I show here in this cartoon that we actually get our stem cells from the adult eye itself. From donor eyes that are coming from the eye bank, we can isolate these spheres of stem cells in a culture dish, on plastic petri dish culture. And these cells, these retinal stem cells, and we say retinal stem cell spheres, they have the potential to transform into all of the different cell types of the retina. And in recent years, we've developed excellent methods at developing very high purity cultures of photoreceptors to replace in the adult retina. And in fact, in this next slide, we see that when we actually transplant these cells into the retina, these beautiful green cells we see are all integrating into that outer layer and developing the nice long processes that are needed to support vision.
And then April, if you could just zoom into these names here, because I just want to take a minute. I want to take a minute to point out and to thank Brenda, because Brenda's helping with the lab tour here today, and it's amazing. I'm so happy because I actually used to work in this lab with Derek Vanderkoy, and Brenda was here then, and she's taught me so much about stem cells. And I also want to point out Molly Shoykit, because it's very cool. Dr. Shoykit is the first Minister of Science for the province of Ontario. So this is such a great, incredible collaborative group. I want to point that out.
Yeah, here at the University of Toronto, we have an amazing collaborative network of scientists working to both understand the stem cell biology behind our new therapies, but also new and innovative ways to transplant these cells using biomaterials like I described. Maybe we can show one of the biomaterials that we've developed. I'll put on some gloves here, because we put a food coloring into the gel that we actually use to transplant into the eye. It's not normally colored, it's a clear gel that we use to inject our stem cells. But we filled a syringe here. Now this gel, this blue gel, it normally has a suspension of stem cells and photoreceptors within it. And we can inject this through a very, very small little cannula into the eye underneath the retina. It's like a needle tip, but it's soft, plastic. And it's what's used during a surgery to actually transplant the cells. Very, very small, thinner than a human hair. And we can inject those cells underneath the retina, suspended within our gel. This gel is it was designed to have very specific properties. This gel, at body temperature, is a solid. It's like a jelly matrix. It has a consistency of what you could think is very, very soft jello. But under some pressure, it actually flows like a liquid. And we designed this gel to have this, what we call shear thinning property, which means that at body temperature, it's a solid, and it can flow into the eye just like a liquid. And this avoids some of the complications that could come with trying to transplant a very large solid scaffold. And so it's our thought that perhaps this technique may confer a lot of benefit for a future therapy in delivering photoreceptors to try to replace them in the retina. If you see, if we inject a little bit out the end of the tip, I'll put it out on here onto one of the other gloves here. We see that it actually, as it comes out, forms little loops of polymer.
That's the HAM-C.
That's right. It's a physical gel.
Oh, wow.
It's flowing like a liquid, but as it comes out, it turns into a jelly. And it turns into a solid. As you see, it's nice, long, ropey bands, and it actually keeps those cells distributed in the space underneath the retina.
So
it disappears entirely within a week.
How many, I know there's not stem cells in there right now, it's just the HAM-C gel, but how many stem cells do you think would be in that amount of gel?
In that amount of gel, hundreds of thousands.
Hundreds of thousands.
But you have to understand that the amount that we would inject underneath the retina would only be a couple of microliters of cells. We're looking to inject typically in our transplants on the order of 100,000 cells.
100,000
in the volume we would use for human transplantation.
I have to take a minute because right now the Foundation Fighting Blindness, we're having an awareness campaign called More to It. Because we're trying to raise awareness. There's more to vision than just losing your sight, more to sight loss than losing your sight. There's more to science, more to research than just sort of the science that you might hear about in the newspaper. And I think the really cool thing that we can learn from you today is there's more to trying to develop a stem cell therapy than just the cells. And that's something that we're seeing with these two most recent papers, which had a lot of bioengineering in them. Brian is actually a bioengineer by training as well.
That's right.
And Dr. Molly Shoykit's also a bioengineering professor. And so it's really exciting to see these collaborations with the bioengineering community that are developing things like gels, scaffolds. I'm trying to think of the language that was used
in the papers. They talked about synthetic scaffolds. We can talk a little more about that later. There's normally two types of gels. There's natural polymer gels and there's synthetic gels. Natural polymer gels draw on what we understand about the normal tissue structure of the human body and what kind of polymers go into making that up, and then uses those same polymers in new and different ways to deliver vehicles. Synthetic polymers mean actually creating novel polymers that don't exist in nature in the laboratory, that may have unique and beneficial properties to deliver stem cells. Those are two different schools of thought in bioengineering, two different schools of practice, and we've worked with both in the lab. Our gel that we found has a lot of benefit as a natural polymer, but we'll talk later about what these two studies have shown they can do with synthetic polymer scaffolds.
There's so much more to it. Wow. Okay, so keep us walking. April, you have to come see this. You know, as I said, it's really exciting to be here. There I am with Brenda so many years ago. Sometimes we like to leave the lab and have a little bit of fun, and that was us on a sailing trip. So those were good times. Because
scientists do leave the lab.
Of course we only talked about science the whole time.
Scientists do leave the lab. Yeah.
Sometimes. Okay.
We'll go this way. It'll be like a walk and talk. We'll do it like.
Okay. Yeah, walk and talk.
Yeah. Did you have questions you want to ask?
We always have. We have so many questions coming in. We have a question from Elizabeth that she's just sent in a question saying, do all the researchers working in this area have PhDs? Can you answer that?
That's a very good question. It's not a prerequisite to have a PhD to work in this area, but most individuals who are studying a specific area of science for a long period of time end up, as they go through their schooling, developing an expertise in that area that allows them to accumulate a body of work to publish and put out there as a PhD thesis. So many of the colleagues we work with have PhDs, but we work with a lot of different people outside of science as well. We're working with clinicians who have MD degrees but have some background in research. We work with people who have master's degrees, and we also have a lot of undergraduate students working in the laboratory who are helping us out with a lot of our projects. So it's a very diverse background to the team. It's not a prerequisite.
And I was thinking, just to point out, like when you first started working on the Ham-C gel, you wouldn't have had a PhD. You would have been in a PhD program.
That's right.
So some of the most exciting and innovative work comes out of young scientists who, really bring in so much creativity and inspiration and hope. Often they're a little less jaded.
They have that idealism which we're looking for, and
which is so important.
We're always looking for new and young talent to enter the lab. People who have backgrounds in science, who have an interest and enthusiasm for learning more about a specific area of the body. We think the eye is where it's at. We think that this is where applications in stem cell research are going to happen first.
Well,
there's great evidence for that in the literature. We're going to talk about some of those papers. And as a young scientist starting out in this field, I was very excited about the potential impact I could have, not only on the field of science, but as a trainee in medicine, also on patient care. And that's what drives me, and I hope inspires young people.
And then Shazad has asked a question. Thanks, also Elizabeth, Shazad, thanks for tuning in. We love our live viewers. Said that, basically, Shazad, I'm going to sort of generalize your question because this is a question we had, tons and tons of questions coming in leading up to the session. And many people are sending in questions about different specific diseases and saying, we see dry AMD, wet AMD. What about these other diseases? So, there's so many different inherited retinal diseases and other diseases, glaucoma. Shazad's asking about Stevens-Johnson syndrome, different syndromes and diseases that cause people to lose their sight. So many of the questions are specific about, what about for this disease? Is there hope for my disease?
Many diseases in the eye follow a common final pathway, and that's that the cells responsible for supporting vision, they die, they degenerate, and they can't be regenerated naturally in the body. Okay, in the adult, they don't regrow. In the retina, it's because this is like brain tissue. Those cells can't reproduce, they can't reform those photoreceptors. In diseases like Stevens-Johnson syndrome, it's because of a severe inflammatory reaction on the surface of the eye that causes scarring, and that scarring tissue won't regenerate into the normal clear corneal surface we need to give us really good vision. And so the idea, the concept behind stem cells is the idea that we can regenerate those cells and put them back into the eye. And so many of the strategies we work in the lab, trying to understand the biology of stem cells, trying to understand how they can be transformed into these different types of ocular cells, is all about addressing those final common pathways in disease. And so we think that our therapies will ultimately have multiple applications across many different diseases. I know today we're focusing on obviously those exciting trials in macular degeneration, but many of our cell therapies, we've also been looking at their applications in diseases of a juvenile onset, like retinitis pigmentosa and other sort of inherited retinal dystrophies. We're looking at them and how they might address patients who have retinopathy related to diabetes, for example. And we've even started working a little bit about limbal stem cells on the surface of the eye and how they can contribute to the regeneration of the ocular surface. And so we're trying to leverage stem cell biology to address all of this disease. It's an excellent question. Obviously, every day in the clinic, I get patients suffering from a lot of different pathology, very diverse. And they all want to know, how are stem cells going to help my specific condition? And I have to tell them that we're working on a final pathway that's going to actually be applicable to many different diseases.
Great answer. So let's get going.
Yeah, we'll do a little walking tour. So our lab is a very large space.
Can you hear?
Can you hear okay? We'll stay close to the camera. Yes. Okay. Our lab is very large. It's a translational space. It's an open concept laboratory, and as we go through, you'll notice just how much space we have. You'll see clutter everywhere. These organized clutter. To the untrained eye in the laboratory setting, this looks like complete chaos. Yeah. But to us, every one of these reagents serves a purpose.
It looks highly organized to me.
We spent a lot of time organizing for your visit.
This is a Canadian company too, right?
That's right.
Yeah,
that's
so
correct.
That's very cool. So just one in the lab. Oh, there we go. Okay.
Are we back?
We're back online.
Hopefully we don't run into any Wi-Fi dead spots.
Okay. This is this is our Wi-Fi
That's
spot.
your hotspot?
We're carrying it around.
Okay. This is a new format.
Here we go.
Okay. So it's a quiet morning in the lab. Also, we cleared space for us on our walking tour.
Thank you.
You'll see as you look down the benches, microscopes, pipette tips, glassware, gloves, lab books to keep track of results and data. These are all the things that go into a laboratory. And all of this, costs money to keep up and maintain, and you have to work constantly to keep it clean. You'll see, as we go, refrigerators, storage for our cells. Cultures area, freezers, fridges, things we need to keep these biological samples fresh. We can keep working with them every day.
Very cool.
Sometimes we have very large pieces of equipment. With this particular piece of equipment, for example, this is one of our pieces of equipment we use for Quantitative Rt-Pcr. That's a very complex acronym, but allows us to look at the expression of genes within our cells. We can understand what those stem cells are producing, what kind of genetic material they're using to maintain themselves as stem cells and to turn themselves into differentiated cells like rod and cone photoreceptors that we're going to use. And by using equipment like this, we can tell how close are those photoreceptors we make from stem cells. How close are they to the actual photoreceptors that we're born with, that we live with? Because what we want to do is, from these stem cells, make photoreceptors that are identical to the ones that we have in our own body, because we want to replace those ones that are lost in disease. So all of this equipment goes into showing us that we can actually do that from a stem cell in a dish.
Yeah, one of the questions that came in from, from Fran, who always asks great questions. Fran was saying, there's so many different kinds of stem cells. Can you give us a little bit more background about, what is a stem cell? And, when I was in the lab, we always used to think about, one of the incredible things about stem cells is you can't just look at it with your eye and be like, that's a stem cell. The way that you tell what a stem cell is you have to do all these kinds of different tests with it, see what kinds of genes are expressed, see how it behaves in a dish. And so part of the work of even identifying stem cells requires a lot of testing.
Yeah, what makes a stem cell is not what it looks like you said, it's what it can do. A stem cell is defined by its ability to self-renew, that is, a stem cell can make more copies of itself, and a stem cell is defined by the other key property of being able to make any other cell type in that tissue of interest or in the body. And that power to make any other cell type is what we're looking to harness in the lab. And that ability to make those other cells is why we have such complicated equipment to try to test that ability, to see how well it's being able to make those other cells, how accurately and how efficiently it's being able to make those cells. And so we often have to very carefully manipulate the culture conditions, the broth that we grow these cells in the plastic petri dish, adding the right factors, proteins and growth factors and things these cells need, things we understand that these cells need from development, what we understand about the development of the human body. We can add those factors to make those stem cells to show their true potential, to become the cells that we need for therapy.
Very cool.
Let's keep walking.
How's our connection? People
Good
here?
connection,
Awesome.
excellent. We'll go around this corner because I want to take everyone to the area where we actually grow these stem cells, where we isolate them, where we culture them, and show you some of the equipment that's in there, and maybe there's some people hard at work.
Cool.
Okay. Thanks. So I'll bring you a little ways into this room. We don't want to disturb anyone who's hard at work here today. I don't know how big it looks on Facebook Live, but we have a couple of what we call Biosafety Hoods here. These are think of them like fume hoods. If you ever remember your high school chemistry class, these are the safety hoods we work under to actually perform our cell culture. You'll see if you come a little further into the room, this is a very clean space. Everything underneath this hood is sterile and clean so that we can grow these cells in a very controlled environment and so that we can keep them pure to understand what properties they have inherent to them and avoid contamination from bacteria, etc.
You can see there's kind of noise, like the air is being sucked up so that there's...
You'll hear that whir in the background. That's the HEPA filters that are working to purify the air that we have. We have stand-up microscopes that we use to count these cells, to see how many photoreceptors we're making, to see how big they become, and to see if we have enough for transplantation.
And then they grow in here, right? This is an incubator?
This is one of our refrigerators. Actually, if you turn around, you'll see there at the far end, one of our incubators. Everyone in the lab, all of our graduate students have a great sense of humor. So usually our cartoons are about avoiding contamination and making sure that we keep our cultures clean and pure for therapy. For example, this one on the door. Things you shouldn't do. Never ignore cell contamination. Reminders to our students.
And I have to show you, April, can you come here for a minute? I mean, one of the things that's amazing, you always see these shots of scientists pipetting, and you can see all these pipette tips in here, and I was reading through one of your plans, Dr. Ballios, about how many pipette tips you were going to use in a year, and it was like
thousands of pipette
thousands
tips.
and thousands and
Absolutely.
thousands and thousands.
Absolutely.
And that's the reason why this work costs so much money to do, right?
It's true. It comes back also to
you can spend like $10,000 on pipette tips.
Oh, absolutely.
So that you don't contaminate your cells.
That's right. And when you think about what we're using to grow these cells, the media, you can see fabulous shots in the popular media and on TV of people working in labs and beautiful pink and green colored solutions. Those solutions are specifically concocted formulations that have a lot of growth factors that are made to actually put in there, and they're made to order. And 100 milliliters of media for us costs $100. It's $1 per milliliter of media. So we tend to get as much out of those cultures as we can because if we have a plate where we're using 200 milliliters of media in a day to feed those cells and keep them growing, that's $200 we're using right there just to keep those cells alive.
Wow.
And once the cells have used it, we can't use it for other cell cultures because of cross-contamination. So
it's worth it.
Shall we make our way back to the conference room? Okay. We'll go this way.
You can see our beautiful city of Toronto.
We're looking across the University of Toronto campus here. There's Convocation Hall. That's one of the great things about working downtown is Look at this beautiful view of
Totally,
this.
it's incredible. And you know, scientists love making posters. It's the way that they communicate to each other. It's like the way scientists talk to each other, poster speak.
That's right.
Yeah.
That's right. So many of these posters have actually been presented at international conferences. And the students and the PhD students and researchers who presented them there, after they're done, bring them back, put them up in the lab, and many times they serve as points for discussion for us to come out and chat about our data.
And I want to say one of the questions that so many of our supporters often ask is, can't you get these people to collaborate and work more together? And so many of the scientists already are working together. So I want to point out, people are often excited about Andrzej Naj's work on age-related macular degeneration of stem cells. So you can see that he's a collaborator here with Derek, and it's really wonderful to see new ideas that come out of these wonderful synergies together.
As you look, you'll see that the breadth of research covers everything from what we understand about retinal disease and what we understand about disease in the eye, all the way to our basic understanding about how stem cells are born in the body, what makes them stem cells, and even further back to our understanding of the development of the human body. Because our understanding of development ultimately informs our understanding of how stem cells turn into the exciting cell types that we want to use for therapy.
If we start talking about developmental biology, we could be here all day.
All day,
this is one
all
of
day
my
long.
favorite topics,
We should
so we're
keep
going to
going.
keep going. Yeah. How's your arm, April? We can't do these Facebook Live sessions alone, and so I want to know that behind the scenes, a lot of hard work is happening. You can't even see the top of the CN Tower out there.
No, covered by fog.
Well, I'm going to keep wearing my lab coat because it makes me feel very sciencey. I like it.
That's great. You should. Today's a day for science. And you were in the lab tour. It's just
That's true. So now I have to let our viewers know that there were so many questions that came in. We're going to try to get to them, but Dr. Ballios actually is off to the clinic this afternoon.
That's right.
So we would love to stay here. I would love to stay here all day answering questions and chatting with Brian, but we will, if we can't get to all of your questions, we're going to do our best to follow up afterwards.
Perfect.
So I wanted to make sure that we could start by talking about these two papers that came in because we're having so many questions about them, and you're really the perfect person to talk with about them because of your bioengineering background and because they're both very much bioengineering papers. So one was published in Nature Biotechnology. This is the phase 1 clinical study of an embryonic stem cell derived retinal pigment epithelium patch. That's the word I was looking for, patch. In age-related macular degeneration. So we have this paper, which was on wet AMD. And then we have this other paper that just came out in Science Translational Medicine that's titled A Bioengineered Retinal Pigment Epithelial Monolayer for Advanced Dry Age-Related Macular Degeneration. And so rather than really diving into the details, which maybe I can do in a follow-up story, why don't you tell me sort of what really excites you about the developments that are being talked about in these papers?
Well, let's start first with the first paper you mentioned. That was the one from Nature Biotechnology. This was the study where this group looked at transplanting that retinal pigment epithelium, that thin single layer of cells that, if you recall on the PowerPoint at the beginning of the tour I showed, sits underneath the photoreceptors and supports their survival and supports their normal function. They looked at transplanting a single layer of those cells on a patch. And what's a patch? Right? We talked a little bit about scaffolds. We talked about natural polymers and synthetic polymers. What this group did, this group that's based out of the United Kingdom, what they did was they developed a synthetic polymer scaffold, a synthetic polymer patch. They used polymeric materials, polyesters that don't normally exist in nature. They actually synthesize them in the laboratory. If you remember your organic chemistry, in a big flask, they synthesize these polymers.
They laid it out and they created a very thin wafer out of that. And it's a solid patch, if you imagine a patch you'd use to patch something on your clothes. And they actually grew those pigmented cells that they made from human embryonic stem cells. They grew them on that patch. And the idea, the concept behind what they were trying to do in their therapy is they said, okay, in wet macular degeneration, what happens? We lose the pigmented cells that support the photoreceptors. Those pigmented cells break down because there's a growth of new blood vessels in wet macular degeneration. And the 2 patients they treated were patients with very severe wet macular degeneration. In fact, they had had a bleed underneath the retina because those new blood vessels are leaky. And so they took those 2 patients to the operating room. The retinal surgeons that operated took away the blood. And because that area of pigmented epithelium in the support layer was destroyed, they took the patch and they put it over top. And so they put that patch on underneath the retina to support, the idea is to support the survival of the film, the retina itself, the photoreceptors that sit over top. If they hadn't done that, well, we know what happens when we leave wet macular degeneration without treatment. After a large bleed like that, we get progressive loss of those photoreceptor cells and progressive loss of vision. Now, their study was primarily looking at a safety study. It's what in the title is called a phase 1 clinical study. What they want to know is, are these cells safe and can they survive after transplantation? Looking at their ability to regenerate vision was not a major focus of this study. The study was not designed to look at that. But what they did see, excitingly, was that at 12 months after they delivered these cells, was that they could actually find evidence with special photography, with special ways of scanning the retina in these 2 patients. They could find evidence that these cells on this patch had survived and that they were still alive underneath the retina. And importantly, that it hadn't caused any of those feared complications that we have when using stem cells. And so what are those? What people have been worried about for a long time with the use of human embryonic stem cells is that they might form tumors after transplantation. And the exciting thing in this study is they showed that in all of their work in the animals before they got to the clinic, and in all of their in the work in these patients, they saw absolutely no evidence of tumor formation. And that's been very consistent across the board with many groups that are working there before getting to the clinic, that they haven't seen any evidence of the ability to make tumors. And they challenged these cells with very specific types of hoops they had to jump through to try to show that not only were there no tumor cells in this transplant, but actually if they tried to put any undifferentiated cells that they're worried about, those cells actually couldn't survive on the graft. And so they, with 2 different very strong pieces of evidence, they demonstrated that their patch is a very safe type of therapy. So those are the really exciting things to come out of this paper. I think it's hard to know what kind of function this is going to give back. These patients had a very severe injury to the retina, a very severe advanced stage of their disease. And they didn't have a comparison to a group of patients that they took the blood away from and didn't put the patch and compare how well they did after just removing the blood. So in science, we have to take all of our results in context. I think that's what this really shows us, is that you can design a study to look at a very specific type of outcome, like safety, but you can't necessarily from that study draw a conclusion about efficacy. And I think
efficacy doesn't work.
Does it work? Is it actually going to help patients see better? That's the subject of their future studies. Those are the subjects of phase 2 clinical studies. So I think a very important hurdle is being addressed here. A very important hurdle not only for applications of stem cells in the eye, but applications of stem cells in general for many different diseases. And they're kind of showing us the way for what kind of things we need to be doing to make these therapies work in people.
That's so helpful. And we're getting to 5 minutes. We only have 5 minutes to go. And so that's why I have to make sure, before we get your kind of insights on this paper, the next one, there's one question that I really must address because it came in from, I forget her, I think it was Suzanne, Wendy, so many people sent in questions. A number of questions came in asking, but what about those treatments that are already happening? Can I go to one of those clinics that's already doing the treatments? And that's where I have to say no, because there are no clinics where there are proven, safe, and efficacious stem cell treatments for vision loss. And we have to reiterate that every time we have one of these sessions, because it's very unfortunate that companies that don't have safe treatments, they don't have treatments that work, but they're pretending that they do. So
you can't jump the gun.
You can't jump the gun.
There are well-established benchmarks for success, and you have to show safety and efficacy through a number of rigorous phases in a clinical trial. And just because something sounds like it should work doesn't mean it necessarily works unless you compare it against people who've had the disease and been treated with the current standard of care. And so this is not a control group. And this paper doesn't even get to that particular stage yet. It shows us just that first basic benchmark. Can we do the procedure, and is it safe after we do it? Does anything bad happen? The answer in this study was, with these 2 patients, they didn't see anything bad happen.
Which is incredible.
Which is great.
Really great progress.
They're enrolling more patients. They're going to keep going. This is very preliminary data, but it's exciting for the field because it tells other researchers who are working with these cells like us, you're on the right path, keep going. You can develop therapies in this space that are already being tried.
Great. I had to get that in because I'm always worried about that.
Me too.
So what about this paper? This one just came out, and this is the dry age-related macular degeneration paper. So what are your thoughts about, again, this is another kind of bioengineering using a scaffold. I'd love to hear your insights.
Again, it's the idea of a patch. It's a different type of synthetic polymer, and you can make any sort of type of polymer patch you like. They again use pigmented epithelial stem cells that are derived from human embryonic stem cells. They grow them on the patch. But as you said, they're using it in dry macular degeneration. And we know the majority of patients with macular degeneration have dry macular degeneration. And many of those patients actually never go on to form wet macular degeneration. And so it's an incredible burden of disease. And unlike therapies for wet macular degeneration, we actually have very few things that we know are evidence-based treatments for dry macular degeneration. We have vitamins, and that's almost about it. And we know that those patients go on and progress in their disease. So it's exciting to see them actually take this concept of patches and translate it into dry macular degeneration, which is going to treat a larger burden of disease across the population, many more patients. And they, again, chose a patient with, or 5 patients in their study, with very severe dry macular degeneration. The idea is that we maybe be able to use this in patients with earlier disease to help to support the survival of the retina.
And that was actually a question that came in from George, who was asking if stem cell treatments could be used to treat both dry and wet AMD. So I think we've got an answer for George.
Yes, I think this is a great example. These 2 papers show that a similar concept is being used in 2 very different forms of macular degeneration.
And Alana sent in sort of a complimentary question about, because I know this is sort of Stargardt, so some of the trials that are looking at dry AMD also sometimes will extrapolate to Stargardt's disease. What do you think about in this case?
I mean, I think that's a great extension of the application. I think that there are some clinical trials going on that are aimed at enrolling patients with Stargardt's and replacing again these pigmented epithelial cells. I think a lot can be learned from these trials about the approach you would use in Stargardt's. Remember, Stargardt's, just like macular degeneration, there's patients very early on in their disease process who still have quite good vision, and there's others who have quite advanced disease. And so it'll be about translating these results, figuring out how to treat patients earlier in their course of disease to prevent vision loss in the future. But again, I think these concepts are movable between different diseases because we're using the same fundamental concept, putting back the cells that are lost to restore the function of what's there.
One more question?
Sure, please, go ahead.
Okay. You know what? I guess the question was just about whether it would work for people who had already lost most of their vision, and I think you've actually answered that. Yeah,
I think in advanced disease, you're putting back the pigmented epithelial cells to help prevent further loss. What we've been working on in our group and in our multidisciplinary team is actually putting back the photoreceptors that are lost in the disease. And so I think from our perspective, that actually represents the application of stem cell therapy that's going to put vision back that's been lost. Most of these therapies, yes, are aimed at stopping the disease process in its tracks and enhancing the function of what's still there. For people with very advanced disease, a lot of the building blocks have been lost. And with our photoreceptor transplantation, we're now reaching out to that stage where we're saying, let's put those building blocks back. Yeah.
Well, thank you so much,
Pleasure.
Dr. Ballios, for welcoming us for the walking tour. It was so fun.
Great.
Thank you to our viewers for tuning in. Thank you to everybody who sent in questions. We had so many questions coming in. I promise to go back in, make sure everybody has the answers. This is one of my favorite opportunities to reach out to people around the world who are tuning in. So we want to keep doing more, and we can't do it without you. So if you've been watching, please like, please share the video, and spread the word, because you know that vision science, there's more to it, and it costs a lot of money. So thank you to all the donors who make research like Dr. Ballios is possible and make the work at the Foundation Fighting Blindness possible too. So happy Friday, everybody. Happy weekend, and we'll see you again soon.
Thanks for visiting.